Hello friends, this is Dr. Pankaj Kumar, your mentor, and in today's sessions we will be talking about virus. We are all aware about the virus and credit or discredit goes to the coronavirus, right? So let's try to understand in detail about the structure of the virus. First of all, virus is a subparticle, or what we can say is a non-cellular subparticle. Why we are calling non-cellular? Because actually you see, this is in between the living and non-living. So it is living only inside the host. That's why we call them obligate intracellular parasite, right? But outside the host, it is a non-living. Okay. And since it is a non-living, it can be crystallized. Okay. And they are living, and since they are living, so they have a genetic material. Genetic material could be DNA and RNA, right? And these genetic material are enclosed outside by a proteinaceous layer. We call them capsid. We will discuss about it in very detail. Okay. So before we move, let's try to learn some historical perspective. So first of all, the virus-like thing came into existence by the effort of the Russian scientist, that is Dmitry Ivanovsky. So he first of all observed that certain microbes are responsible for causing a mosaic-like disease in tobacco. Later it was Byjernik who observed that when this virus or when this infected particles are taken and is transmitted to the fresh leaf of tobacco, they also get infected. The name virus in fact was coined by Byjernik. And finally, it was Stanley who crystallized the virus. And crystallization of the virus means that virus is a non-living, because only non-living things can be crystallized. Right? Let's talk about the shape of the virus. And in order to give you an idea about the shape, I would like to show you this comparative diagram of the different cells. Have a look at the blood cells. So very big in shape. Then the E. coli. So these two cells will give you a very clear idea that what are the sizes of the virus. So for instance, the adenovirus, the bacteriophage, these are very, very small in comparison to the bacteria or the blood cells or the normal cells, right? Now let's talk about the virus shape. So it is of different shape. For instance, it could be helical, right? It could be polyhedral, just like influenza, or it could be complex like bacteriophage. You can see in the diagram as well, right? So these are the different possible shape of the virus, okay? Now let's talk about the structure. It is a nucleoprotein, right? So there is a genetic material, and outside the genetic material there is a proteinaceous covering, and that proteinaceous covering what we call capsid. Genetic material could be RNA or DNA. Popularly we know that in case of plant virus it is a RNA. In case of animal virus, what happens? It is a DNA. Okay. So virus infecting plants generally have a single stranded RNA, and those animals we know very well that is a double stranded DNA. Okay. Now there is another category of virus what we call bacteriophage. Now these bacteriophage are those viruses that attack bacteria. We have also discussed about bacteriophage in the chapter of Monera when we are discussing about transduction. This was one of the means for the genetic recombination, right? Now you see, the outer coat is a protein that is called as a capsid, right? And this capsid is individually made up of smaller unit what we call capsomere. Okay. So these capsomere are arranged in helical or polyhedral geometric forms, right? And the very function of the capsid is to protect the inner core or the inner genetic material. Now we know very well virus cause diseases. So viral disease could be mumps, smallpox, herpes, influenza. So these are the viral diseases. One of the very common is the AIDS. The AIDS is actually caused by the virus, right? And in plants, again, can be large number of viral diseases, right? And the symptoms could be mosaic as we observed in case of tobacco, right? Then we have a yellowing of vein, right? Dwarfing, a stunt growth. All these things are going to occur due to virus. Let's discuss what are the treatment for the virus. You see, a vaccine against virus can be made, and these vaccines are made by the attenuated virus. Now, what is the attenuated virus? We take out a protein, we just remove the infection causing part, right? And then that attenuated virus is injected. By that, our immune system is triggered, and against that immune system is going to form the antibody. So that's why the viral vaccine is actually being created, right? Now, against the virus, our defense mechanism is going to form one protein, what we call interferon. It is just like antibiotic, which is synthesized or formed to kill the bacteria. So for bacteria, this is antibiotic. Similarly, for virus, it is an interferon. At the same time, there is a certain antiviral drugs, and one of the most common popular is AZT, right? That is azithromycin, right? That is used to delay the progress of the AIDS. Okay, and in addition to that, there is a protease inhibitors as well, right? And they prevent capsid formation. So these are the ways in which the viral diseases are actually being treated. Let's talk about the bacteriophage. This is the most studied one, and we have already discussed. This is a virus that is going to attack bacteria, right? So what happens? One of the most studied bacteriophage is the T phase, right? So T phase has a double-stranded DNA as a genetic material. Outside, there is a protein code. We already know that that protein code is actually called as a capsid. The most commonly studied T phase, what we call T4 and T7, okay? And they infect E. coli. That is the E. coli we know very well. That is our intestinal bacteria. So have a look at this diagram. Okay, so this diagram will give you a very clear idea. You know here, there is a head, right? And within the head, there is a genetic material. Then we have a neck, then we have a collar. And there is a small spike over there. So that spike is actually being utilized to penetrate the bacterial cell. So they inject DNA to the bacterial cell, and later on that bacteria is actually under the control of the viral DNA. So there are two different types of cycle, what we call Lytic and Lysogenic cycle. We will discuss in detail about that, right? So have a look at virus life cycle. Viruses are host specific. So if you're talking about one particular virus, they infect only the given host. So they are not going to attack the entire set of the living beings, right? So there are two types of viral cycle, what we call Lytic cycle and Lysogenic cycle. Now, what is Lytic cycle? Lytic cycle is one when the virus attack the bacteria, right? And the bacterial cell will eventually get destroyed or destructed. This is called Lytic cycle. And what is Lysogenic cycle? In case of Lysogenic cycle, what happens? That virus get integrated with the bacterial chromosome. And once they integrate it, they keep on multiplying. Later on at some stage, what happens? They can undergo the Lytic cycle. Okay. So when the viral genetic material replicate along with the bacterial DNA, we call it the lysogenic cycle. Okay. So there are different stages for the Lytic cycle. For instance, the very first stage is attachment, where bacteriophage attach with the bacteria. Then we have a penetration. The penetration is going to happen by the spike-like structure, and there is a one enzyme called lysozyme that opens the cell wall, right? And by the contraction of the tail fiber, the DNA is injected into the bacteria. And once the DNA is injected, then there is going to have a biosynthesis of the viral DNA, and that viral DNA is synthesized while taking control of the machinery of the cells. They use the entire enzymes that is required for the DNA synthesis of the bacteria, and more viral DNA are going to be synthesized. Eventually, protein are also synthesized, that is viral protein are also synthesized, and the new viruses are actually being formed. This is what we call maturation. And finally, once the large number of viruses are within the cell, what will happen? That they will just destroy the bacterial wall, and all the new viruses are going to be released, right? So this is the popular Lytic cycle. Okay, have a look at this animation. So this will give you a very clear idea. So this is the first one, that is the attachment, right? The viral bacteriophage is actually attached with the bacteria. Now after this, what will happen? They will penetrate. You can see there, after the penetration, what will happen? That the viral DNA is coming inside, right? And once the viral DNA comes inside, what it will do? It will start making its own copy. You can see there, more than one type of DNA are forms. So viral DNA are being synthesized. Then the virus get assembles. And once the virus get assembles, what happens? They will break the bacterial wall. And once the bacterial wall is breaking, the new virus are going to be released. This is what we call as a Lytic cycle, right? I hope this animation will give you a very clear idea that how the Lytic cycle runs about. Okay. Now let's talk about the lysogenic cycle. Now in case of lysogenic cycle, what happens? that the virus, after injection, they do not break the cell, rather they get themselves integrated into the bacterial chromosome. Okay. And once they get integrated into the bacterial chromosome, with the bacteria, they keep on replicating, right? And the injected part of the virus in the bacteria is actually called as a prophage, right? And as I told you, along with the bacteria, they keep on replicating. So over the period of time, large number of prophage, that means the viral DNA is going to be formed, okay? And once a prophage cell is activated, again, the same host cells will enter into lytic cycle, and it will follow the same thing what we have seen in the previous cycle. So again, under the lytic cycle, the host cell is going to be destroyed. So this is all about the lysogenic cycle, right? So let's see this diagram. So that will give you a very clear idea about the comparison of lytic and lysogenic cycle. On the left hand side, you can see there is a lytic cycle. On right hand side, you can see there is a lysogenic cycle. So in case of lysogenic cycle, you can clearly see that the red colored structure, that is a viral DNA, right? They get integrated into the blue color structure, that is the bacterial DNA, and they keep on replicating. So once the inserted red color structure within the bacterial DNA, this is what we call prophase, right? So as we have discussed, so once the prophase gets activated, they will enter into lytic cycle, and eventually they will destroy the bacterial cell, right? So this is about the two types of life cycle in case of virus. Okay. Now let's discuss one more thing. What is viroids? You know, viroids are an infecting agent that is made up of the genetic material, that is a circular RNA, but they don't have any protein code. Protein code is absent. That is the most important thing. And this is an infectious particle. Okay. So RNA of viroid is of the low molecular weight, right? And it causes potato spindle tuber disease, right? And it also responsible for the potato famine of Ireland, the famous potato famine, famine of Ireland that is going to occur due to destruction of the potato crop, right? So that was about the viroids. In some way, it resembles introns cut out of the eukaryotic cells, right? So you can see there. So that is the structure. So this leaf is actually infected by the viroids. Okay. Let's talk about prions. Now, what is prions? Prions are infectious proteinaceous particles. They don't have any genetic material, right? So what happens? These proteins are abnormally folded structure, and once they come in contact with another prions, they also get folded. They don't have any genetic material. They are only proteinaceous particle, right? And they cause diseases. Okay. So what happens? In case of humans, they form an insoluble deposit, which eventually lead to degeneration of the neurons in case of humans. And one of the most well-studied disease that is caused by prions is a mad cow disease. We call them bovine spongiform encephalitis, right? That is caused by the prions. Okay. So it is also analogous to Jacob disease that is found in case of the humans. So that's all as far as the entire virus is concerned. Now it's time to test yourself. So this session is going to be followed by some questions, and these questions is based on whatever the things right now we have discussed. Please go through those questions. In YouTube, these questions may not be visible. So I suggest all of you to register yourself in the link that is given over there. So in these links, you will find the videos along with the questions, right? In the next sessions, we will be talking about the protest. Join me in another session of the NEET Bite, right? Thank you.